Welcome back to Chapter 5, where we're going to discuss selective permeability and how things cross a membrane directly as opposed to going through a little helper proteins or some other means. So again, membranes or the structure of membranes really helps in this selective permeability. I defined that for you earlier in the previous lecture in Chapter 5. A cell has to be able to regulate the movement of things in and out of the cell. It has to be able to do that in order to maintain its life. And so that is the job of the plasma membrane. Specifically, and this is important, hydrophobic, or those nonpolar molecules like hydrocarbons, those can dissolve in the lipid bilayer, which means that they can cross that lipid bilayer very easily. If you think about it, the nature of that phospholipid bilayer, the vast majority of that phospholipid bilayer is made up of those hydrophobic tails. You can see here that really is, those tails are much bigger than the hydrophilic heads. So hydrophobic molecules can easily kind of slip in between these heads and then hang out here for a little while, slip in between here, and then go to the other side. They can go back and forth very easily. Polar molecules, like sugars, do not cross the membrane easily. And here's the thing. We're going to be talking about osmosis in a minute. Even water does not cross that membrane easily, especially when we compare it to non-polar molecules like we talked about hydrocarbons. We might mention oxygen or carbon dioxide. Those are also non-polar, and they also cross the membrane pretty easily because they're non-polar. So if they're not crossing it directly, right across the phospholipid bilayer, they do go through often by using transport proteins. Transport proteins allow the passage of hydrophilic substances like sugars, like amino acids. Some transport proteins, which are called they're called channel proteins. Let's just bold that for you. They have a hydrophilic channel in the center, and that allows ions to go through it, kind of like a tunnel. So that's kind of nice. Specifically, if we're talking about water, the channel proteins involved in allowing water to go through are called aquaporins, literally water pores. So aquaporins. Again, we'll cover that more later, but we are going to focus for a little while just with the concept of osmosis, eventually not thinking about aquaporins. But we'll bring them back in again as we move along. So we have other transport proteins called carrier proteins. Let's highlight that one too. Carrier proteins are transport proteins that actually bind to molecules, and they kind of change shape. And that changing of the shape of this protein molecule, kind of like, you can think about it as like kind of squeezing a ketchup packet, right? The carrier protein is going to change shape and shuttle things across the membrane. It only does this with specific substances. So a ketchup packet's not going to move mustard across the membrane, right? It's only going to move ketchup across the membrane. So you can kind of think about it like that. So some basic concepts here for you about how things move directly across the membrane. The first word that we're going to learn is the word passive transport. Passive transport means that there's nothing helping it. It's just moving through the process of diffusion. And while some transport proteins might be involved, again, we're not going to think about them just yet. We're just going to think about how diffusion works for now. And diffusion, as I mentioned a little bit earlier, is how molecules move from a place where they're really highly compacted into a place where there's lots of available space. So we say they move from an area of high concentration, like you see here, to an area of low concentration like you see over here. So they're going to be moving back and forth. These molecules aren't just like parading across the membrane though, and the membrane is indicated by this dotted gray line. They're not just like parading across. These molecules, non-water molecules, in this case we're looking at some dye molecules, they're moving around randomly. And some of them happen to bump into there, and then they're going to happen to move around here. They're all just moving randomly. But the thing is that they move randomly, and it's in a very organized fashion. They end up being in equilibrium. In other words, they end up having just about as many molecules on one side of the membrane as they do on the other side of the membrane. So that's equilibrium. If you have two sets of molecules, one on one side of the membrane and one on the other, they're both going to move back and forth until they're both in equilibrium with themselves. So that's really important to think about, because the yellow molecules here are not in equilibrium. They're going to diffuse this way. The purple ones are going to diffuse this way until they're each at their own equilibrium. Another way to say equilibrium is dynamic equilibrium, and that's just saying as many molecules move across the membrane in one direction as another. In other words, those molecules are always moving around. So I've used this word moving down their concentration gradient. I've talked about that a couple times now. So using this word moving down their concentration gradient is really just saying they're moving from where they're very concentrated to where they're less concentrated. And and again, it's not affected by the presence of other substances. Each of those separate types of molecules are going to move down their own concentration gradient. Diffusion across a membrane is called passive transport because there's no energy involved in the process of this happening. There's no energy used up in this process. A very specific kind of diffusion is called osmosis. And I think most of you have heard of osmosis. It is an interesting and challenging concept, but once you wrap your head around it, it's pretty straightforward. It's just diffusion, but it's diffusion focused specifically and only on what happens to the movement of water molecules. And of course, water is so important for life. We have our own word to describe its diffusion. Also, very specifically, osmosis is talking about water moving across a selectively permeable membrane. Remember from earlier that in living things, water doesn't move across the membrane very easily. We have to have aquaporins. But still, it is passive transport, and it is just going to move down its concentration gradient. So what that means is that water moves down its concentration gradient from an area of a region where there's lower solutes to an area where there's higher solutes. This sounds backwards, but it's not. Hold on to your horses as I explain how this is actually water going down its concentration gradient. But we never talk about the concentration of water. We always talk about the concentration of solutes. And so it's going to seem a little backwards until you get the hang of it. If you want more help with this, you can watch the BioFlix animation on the publisher's website, and it will step you through osmosis. So let's take a look at this picture, though, and see if we can get a handle on it. Okay. So here is a U-shaped tube, and in the middle of it is a selectively permeable membrane. On this side, you can see that there's not very many solutes, the green dots, and on this side, you can see that there is a lot of that solutes. Now, because we have this selectively permeable membrane right here, it is not allowing the green solutes to diffuse down their concentration gradient. So that's what we're saying here. Maybe they're too big to fit through the holes in the selective permeable membrane. Here you can see that those green ones are just too big to fit through. And so and they're actually surrounded by water molecules. So this side has way more of those solutes. and they can't go through the membrane. So what happens is the water goes down its concentration gradient, and it basically flows towards the right. When this happens, the concentration of solutes becomes equal. There's just as many solutes on this side per water molecule as there are on this side. We've reached equilibrium, but it looks like this side is much higher, and it is. And that's because the water has gone through osmosis. It's moved from one side of that permeable membrane to the other side of the permeable membrane. Again, if you're having a little trouble with understanding this, please go check out the animation available on the publisher's website. Okay, so water balance is extremely important to cells. It's important to cells that have cell walls and important to those without cell walls, but the way we think about it's a little bit different. So I'm going to give you a few words here, and then we're going to look at how these water balance affects things with cell walls and things without cell walls. Okay, so our first word is tonicity. And tonicity is the ability of a surrounding solution to cause a cell to gain or lose water. So we talk about the tonicity of the liquid, the solution, but specifically with regard to the cells that happen to be nearby. In an isotonic solution, the concentration is the same inside and outside. So if the solute concentration is the same inside of a cell and outside of a cell, there's going to be no net water movement across that plasma membrane. Of course, there's going to be water moving across the membrane, but it's going to be equal. It's going to be in dynamic equilibrium. The next term is hypertonic. In a hypertonic solution, the solution concentration is greater outside the cell than it is inside the cell. If the solution concentration is greater outside of the cell is going to lose water, because remember, the solute's not able to move into the cell. So that means water has to leave and go outside of the cell to make it in dynamic equilibrium. Hypertonic causes cells to lose water. So one mnemonic that you can think about is if you're feeling hyper, if you're feeling really, really active, you might want to get outside. So water, right, is going to leave the cell. If you're feeling active, you want to go outside. In a hypotonic solution, the solute concentration outside of the cell is less than the concentration of that same solute inside the cell. And because of that, the cell is going to gain water. Water is going to move across that semi-permeable membrane, and it's going to cause that water, that cell to expand by water moving inside. So we can take a look at some of the consequences of that by looking at this figure here. In an animal cell, and this is a cell that does not have a cell wall, right? So your cells, all animal cells, lack a cell wall. And in a hypotonic solution, remember that's the last one that I mentioned, it's where the solute concentration is on the outside of the cell is less than the solute concentration inside, and that causes the cell to gain water. When that happens, cells burst, or the word that we use for that is lyse, Lyse. So in a hypotonic solution, a red blood cell like you see here is going to lyse, or it's going to burst, because so much water is moving in that it basically explodes like a balloon. In an isotonic solution, the amount of water that's going in and out is equal in dynamic equilibrium, and so that cell is going to maintain its shape. In a hypertonic solution, that cell is going to shrivel. It's going to, you can see here, it looks like a little starburst shape. It looks that way because it's completely losing its water to the surrounding material. Of course, there are some strategies that cells have to deal with being in hypertonic or hypotonic environments. And any process by which a cell deals with this is called osmoregulation, literally regulating its osmotic or osmosis or water process of moving water inside and out. So one very cool example is seen in the protist Paramecium caudatum. And Paramecium caudatum this is like your typical paramecium. It's a protist, and it has this really, really cool vacuole called a contractile vacuole. We talked about this in the last chapter. Paramecium live in pond water, and pond water is hypertonic, right? There's not very many solutes in the pond water compared to like the salt watery nature of something that lives inside a cell. So that means that water is going to be constantly moving into the cell. Well, this contractile vacuole just basically pumps the excess water out of the cell. It's figured out this very, very cool way of osmoregulation. There are very nice ways that cells with cell walls can deal with water regulation that we animals, with our animal cells, with lack of cell walls, don't have. So cell walls really do help maintain water balance. If you put a plant in a hypotonic solution, that solution, it's going to swell. It's going to cause the plant to swell until that plasma membrane bumps up into the cell wall. And when it bumps up into the cell wall, the cell is something called turgid. That word right there is turgid. That means the cell is very, very firm. It's being pressed up against by the inside. The plasma membrane is just pushing up against it. And it's kind of reinforcing or giving firmness to that cell wall. If a plant is put in a surrounding that is isotonic, that means there's no net movement of water. And when this happens to a plant cell, it's actually not such a good thing. The plant cells become flaccid or limp, and the plant may wilt. You can think about it as putting a very light amount of salt in some water and watering a plant. Your plant is going to become limp because of it being in an isotonic solution. In a hypertonic environment, plants are going to lose water actively. And when that happens, the plasma membrane pulls away from the cell wall, resulting in something called plasmolysis. Let's take a look at what happens in each of those situations. In the hypotonic solution, that's where there's very few solutes, the water is going to be moving in, and basically we have a turgid cell that's very, very happy. In an isotonic solution where there's a little bit of salt, right, a little solutes in that water, the plant becomes flaccid, and for a plant, that's not so great. In a hypertonic solution, look what happens to the plasma membrane. It also shrivels, just like it's shriveled with the animal cell. But in this case, the parts of the plasma membrane are attached to the cell wall there. And so we say that's plasmolyzed. So that's all I've got for you for part 2 of chapter 5. Next video we're going to be looking about how things are transported into and out of cells instead of just going directly across the membrane.